In this video, we are going to focus on the study of pharmacodynamics and toxicodynamics in terms of tissue-level cell signaling. The learning objectives for this lesson are to describe how a drug or toxicant can elicit a response from a cell and result in a whole-body response, indicate ways cells can communicate to each other, and tie signal transduction into the properties of a ligand, including the affinity, potency, and efficacy of a drug or toxicant. Let's do a quick review. Drugs travel through the blood to reach target cells, which have specific receptors on the surface based on their cell type. These xenobiotics can act as ligands and bind to receptors to initiate a response inside the cell. Based on their physicochemical properties, ligands carry information to determine which receptors it can bind and activate for a cellular response. If an agonist tries to bind the wrong receptor, no response will occur, while if it reaches the right receptor, a response is initiated. The response and its amplitude depend on the concentration of ligand at the receptor, the affinity of the ligand to the receptor, and the efficacy of the ligand. How a ligand and a receptor communicate is based not only on the physicochemical properties of the ligand, but also the structure of the receptor. Affinity describes how well a ligand can bind a certain receptor. The higher the affinity, the stronger it binds, which may lead to a longer and stronger cellular response. Efficacy describes how well a ligand can trigger a response. As we discuss these terms in more detail, you will see a drug can bind with high affinity yet still trigger a relatively weak response. Let's think about a drug that targets the lungs and reduces inflammation. What do you think it will do when it reaches the heart or other organs? Maybe it elicits a completely different response on the heart, such as increased heart rate. In this way, an asthma medication may cause unwanted side effects, or a known liver toxin may cause kidney cancer at low doses. To determine the health benefits and risks of drugs, it is important to consider the mechanism of action and how it may affect each organ at several concentrations. To understand the mechanism of action of drugs and toxicants, it is important to know how cells communicate. There are several ways cells can communicate depending on the cell type and proximity to other cells. Let's start with looking at the secretion of molecules. Molecules can be water or lipid soluble. Water-soluble signals bind to extracellular receptors to trigger a response in the target cell, while lipid-soluble signals can travel through the lipid bilayer to bind intracellular receptors, often with the help of carrier proteins, such as albumin. Juxtacrine signaling occurs when two cells are very close to each other, and the signal on one cell surface binds a receptor on a neighbor cell. Paracrine signaling occurs when cells are close but not in direct contact. This includes autocrine signaling, which is when a cell releases molecules that act on its own receptors. Paracrine signals are short-lived. Neurons use synaptic signaling to move signals long distances very quickly and target very specific cells. Often the neurotransmitter is degraded or taken back into the nerve cell, tightly regulating the duration of a signal. On the other hand, endocrine signaling widely disseminates the signal, reaching all targets via the bloodstream. This can be a slow process, lasting up to days. Here is a link for a fun animation on cell signaling to see a full visual of how cells can communicate. But how can a ligand binding to a receptor create a response in the whole body? If you look at the process of cell signaling, you can see it is a many-step process. First, you need a ligand to reach the correct target cell. Then the ligand needs to successfully bind a receptor. The rate of ligand binding and dissociation from a receptor regulates how fast a signal can be induced. This relates back to the affinity of a ligand. If a ligand binds, a signal is initiated. The strength of signal relates back to the efficacy of a ligand. This can lead to the accumulation of second messengers, such as cyclic AMP or intracellular calcium, which can interact with other signaling pathways to change a cell's phosphorylation state, gene transcription levels, protein degradation rates, and many other cellular processes. Changes in cellular function lead to an overall tissue response from a drug or toxicant, which can then trigger a whole body response. We are going to discuss these steps again in terms of affinity, potency, and efficacy of a ligand, so we can better understand dose-response relationships. For a quick review, affinity is defined as how well a ligand binds a receptor. Efficacy is how well a ligand elicits a response, and a new term, potency, is how much of a ligand you need to get a desired response. To discuss the affinity, potency, and efficacy of a ligand, we need to define a few terms. This will be a lot of information, but how we think about a response between a ligand and a receptor is similar to how we think about a substrate and an enzyme and enzyme kinetics. Once an administered drug reaches a cell, there is a given concentration of ligand and receptors. Ligands bind receptors at an on rate called K+1 and unbind receptors at an off rate called K-1. Based on the conditions in or around a cell, there is an equilibrium between free and bound ligand, which can be discussed using the dissociation constant called Kd. Kd is the rate at which a ligand unbinds a receptor. Therefore, a low Kd means the ligand does not leave the receptor easily and has a high affinity for that receptor, which in turn results in a very potent response. Now that the ligand is bound to the receptor, a signal is initiated which integrates with other cellular signals to create an overall cell response that is transmitted to the whole body. This response is called Q. A ligand's intrinsic efficacy describes how well a ligand triggers a response once a signal is initiated. In terms of therapeutic drugs, the clinical potency reflects the amount of drug needed to produce a certain level of therapeutic response. The affinity of a drug for a receptor is the principal determinant of clinical potency. The intrinsic efficacy reflects the greatest clinical response attainable with a drug at any dose. Drugs can be very potent to a receptor but have low intrinsic efficacy. Here is a diagram we discussed in the cell-level dose-response video. Let's talk about full, partial, and inverse agonists in terms of their affinity and efficacy. Full agonists tend to have high affinities for receptors and high intrinsic efficacy, resulting in a robust response similar to endogenous ligands. Partial and inverse agonists may have a range of affinities, but partial agonists always have lower efficacy and potency than full agonists, and inverse agonists always have negative efficacy compared to full and partial agonists. Antagonists, on the other hand, can have high or low affinities but always have no intrinsic efficacy. If a competitive antagonist has low affinity, then it is more likely to be replaced by an agonist with a higher affinity. If you are designing a drug, how would you use information about affinity, efficacy, and potency to your advantage? It is important to note that you need to think about the pharmacokinetics and the pharmacodynamics when evaluating a drug. If a drug is quickly metabolized, then you may need to administer more to reach the same potency of a drug that is metabolized slowly. Maybe the response you want requires a decrease in a second messenger, and if you inhibit G protein-coupled receptor and a calcium ion channel, you get an exponentially greater response. But you need to know how one drug may impact the clearance or distribution of another drug. Here are some more questions to help you think about how ADME and pharmacodynamics both play a role in pharmacology and toxicology.